Well, then, hello to everyone that is here. I want to introduce to you Gemma. She's here as a representative from the Ersilia team, and she's a molecular biologist by training. Her expertise encompasses a wide range of techniques from 3D cell culture to genome editing with CRISPR-Cas. And she holds a PhD in the field of colorectal cancer. And she got her PhD in Barcelona. And she has recently moved to the computational pharmacology field. And she is one of the co-founders of Ersilia, where she's putting her expertise to the development of machine learning models for drug discovery. So, Gemma, you can start with introducing us your work.
Yeah, thank you, Artemis. Nice. So, thanks everyone for coming or watching this recording later if you are. I'm just gonna share my screen. I will try to be not too boring and not too technical for everyone. So, thanks to Trend in Africa for organizing these webinars and letting us access their network of contacts. I'm Gemma, and I will be presenting our small project, which is called Ersilia. And we are trying to do we are doing a hub of artificial intelligence tools dedicated to drug discovery. So I guess the most important is who are we? We are a small team. We started this initiative in September 2020. So Eduardo is also molecular biologist by training. He got his PhD in Cambridge. Michael is a computational pharmacologist, actually a chemist, and he got his PhD in Barcelona and then continued on the field of machine learning and artificial intelligence as a postdoctoral researcher. And myself, as Artemis has said, I'm a molecular biologist by training and have recently moved to the computational field. So our mission in Ersilia is to strengthen the research capacity against infectious and neglected diseases. Why is that? Well, the numbers are pretty overwhelming. As you know, research costs are really high. It takes billions of dollars to develop new drugs and up to 12 years. And this makes research prohibitive in low resource settings. So if we take in as a research output the number of publications, you can see that in low and lower middle income countries, they don't amount to even 10% of the publications in 2018. And this landscape is not likely to change soon because also the number of high-level institutions such as universities is much less in those countries. This leads to a Western bias in biomedical research, as we call it. So as you know, high and upper middle income countries have a burden of disease mostly due to non-communicable diseases. But in low and middle income countries, this burden is much more shared between communicable and non-communicable diseases, despite the fact that many people in the world are affected by communicable diseases. When we look at the drug discovery scene, we see that 87% of the drugs in development pipelines are for actually non-communicable diseases. So that there is a large gap of diseases that are not being addressed by the large pharma companies. And not only this, but as the WHO already stated in 2013, so I just quote them, all countries need to be producers of research as well as consumers of it, because some questions may need local answers and the research has to be adapted to the needs of each country. So if it's not only the type of diseases that are investigated, but where are they being investigated? Our approach to this big problem, our small contribution. So, you know, we are a small team and we think that where we can make the most impact is by applying artificial intelligence models in the drug discovery pipelines. Artificial intelligence models, you may know, is the new promise for drug discovery. So artificial intelligence, well, computer-based tools allow you to screen a larger number of molecules and do it in less time than with conventional experiments. So therefore, improving the drug discovery paradigm. It has certain limitations though, which are basically that most of the research that has been done at the moment is not reusable, either because the code is not shared or when it is shared is not actually easy to understand for the other scientists. There are no clear benchmarks yet because it's a very new technique and we lack still experimental validation for many models, which renders artificial intelligence models black boxes and makes this artificial intelligence research not fully trusted by many experimental scientists. And on top of that, we still require strong technical skills that are not available everywhere, and not everyone has access to the computational capacity to perform artificial intelligence studies. If we go a bit into more detail on how can artificial intelligence help in the drug discovery, you may be very familiar with this pipeline. So the drug discovery pipeline begins by identifying some targets, some genes that are affected in your disease, then trying to identify molecules, leads that will modify these genes, optimizing those leads so that they will have the best pharmacokinetic properties and toxicity properties, finally moving on to clinical trials. And when you have arrived here, if you succeed, you still need to monitor the progress of your pharmac for a couple of years after approval. So this is a very long and costly process, and it's already been proposed that artificial intelligence can be plugged to many of these processes to make them faster and cheaper, such as understanding which genes are more important in the diseases so that should be targeted, improving the leads, so optimizing the chemical libraries from which we develop our drugs, or understanding better the drug-drug interactions and the possible toxicities we have. This realm is already invaded by several big companies. So here, for example, you may have seen this film in Netflix about AlphaGo, which was the first computer that was able to beat humans playing this game, the Go game. So the same team, DeepMind, has developed what they call the AlphaFold, which is an artificial intelligence tool that is able to fold proteins solely using the amino acid sequence. They are able to predict the folding of the structure with higher reliability and less time than any other procedure so far. And this is already a great advancement in research. As you know, crystallizing structures is very expensive and time costly. Also, here you see this painting. Is actually not a painting done by humans, but it's the first painting done completely with a computer. So you may judge the quality of it, but but at the same time, the same manner that now computers can paint like humans, computers can also create new molecules. So I want to highlight the partnership between this data-driven drug discovery company, Exscientia, and this Japanese pharma, because they together have started the first clinical trials using a drug completely developed with artificial intelligence. So there are advancements in the field, which is great, but the question is, how is this applied to academic research worldwide? As a scientist, what can we do to tackle this, to use these new techniques and apply them to our research? So this is exactly what we are trying to do with Ersilia. Our goal is to provide ready-to-use artificial intelligence models. So, for example, if you have a drug or a number of drugs and you want to understand which targets, which genes are affected by these drugs, you can actually use artificial intelligence to do that. But the process is quite complex, so you need to start by having some data. This data needs to be processed, and then it can be fed into the models that we need to train. Once this model is trained, it needs validation, first with our own sets, and then with sets that the model has not seen before, and even possibly we need to validate that the model is working experimentally. So most of the models that are published now in artificial intelligence journals stop here, which makes them highly inefficient to be reused because many people just cannot understand how to use a model. So what Ersilia is doing is adding this extra layer, which is what we call the deployment, to make it simple and easy for scientists to just input a question and obtain a result. As an example, I have chosen a few of the models that we are actually including in our hub now. One of them is just collecting directly from the literature. So this paper came out last year, was published in Cell. It's the largest study about broad antibiotic discovery of broad antibiotic activities. It's great, but the code is fully open source, but of course you cannot, there is no app to fastly use these models. So what we have done is bundle this code. So now in our Ersilia Hub, what you can do is you just write the drug you are interested in, and it will give a prediction of whether this drug is active or inactive. We can go further than this, and we can basically, instead of taking models that are already developed, we can look at data sets. So for example, in this paper, they did a large screening of over 50,000 compounds against for antimalarial activity of liver stages of the parasite. And they discovered a whole new chemical series. So what we have done is taken this data set, and then using that data, we have trained an artificial intelligence model so that now, if you're interested in seeing whether your molecule has antimalarial activity or not, you can just write your molecule, and it will predict whether it's an active or inactive antimalarial drug. Importantly, we do more than that. So we are really interested in finding collaborators so that have their own projects and their own scientific questions that can be solved or partially solved using artificial intelligence to develop specific models and help you or them to answer them. So this is how the model is actually looking. We have a collection of models, each one with its web application. It's not yet released. It will be released next month. We hope to reach 100 models by September. And so now if we take a step back and look at how we are actually organizing these models, so how do we understand the biological complexity of the diseases? We have started here, so we are now focused on drug discovery. So we are interested in seeing how compounds can affect the genes, but we are also interested in all these other relations here. So we always try to place our models to organize them in this blueprint. So for example, we can see how pathways are modified in different diseases, how cells associate in tissues, and this modifies the gene expression, and therefore how this affects diseases and how it relates to the compounds, and so on and so forth. So there is all these different edges or relations that we can actually model. As I said, we are starting with the drug discovery. So the type of models that we have for the moment in our hub are based on compounds. So we understand this compound cell sensitivity, or how they interact with proteins, even how they interact with other compounds, or which kind of toxicities they produce in the body. All these models are free to use. They are available online. And when we have used data or models trained by other authors, we always credit the original authors of the model. And importantly, we try to maintain our models state of the art. And I hope in the next few slides that are a bit more technical, but not too boring, I can convince you of why, what technology are we using to make our models. So the technology we use is called the chemical checker. It's a resource that was developed by Miquel, one of our co-founders, at the laboratory of Patrick Alloy at IRB Barcelona. It was recently published in Nature Biotechnology. And basically what this resource does is encode the chemical information and biological information of small molecules. So you know, when we want to train a model, we have the compound, and we need to give this compound in a way that the computer can understand, so in a numerical vector. Yes, so here we have these chemical characteristics, and we try to encode them here with, let's say, ones and zeros to feed this information into the computer. But actually, this is not the only information we have about this compound. We actually know that this compound is an antimalarial. We know which activity it has. We know its side effects. We know which family of molecules it is related to. So we have a lot of information that in the classical machine learning models, it is not encoded. So what Miquel and Patrick and his team tried was to and to use all this information and put them into vectors so that they can fit much more information into the machine learning models, making those predictions much more accurate. They organize the biological space in 5 different levels. So we have the chemical information about the molecules. We also have, if there is information, what are these molecules targeting, how they interact at the level of networks and the level of cells, so gene expression or the results in cellular bioassays, and even if there is available information, what are the clinical effects, the indications, other side effects or drug-drug interactions. The chemical checker is currently encompassing 800,000 molecules, so it's the most comprehensive resource to date. And using this tool, actually, they were able to train at the group of Patrick a tool that has been top performer in the Dream Challenge last year. And so it is a lot of information behind it. You can ask Mikel, I think he's here in the chat if you have any questions, but the summary behind all these years of work is that we are now able to produce signatures that reflect accurately not only the chemical structure, but also the information we have at the cell level and at the therapeutics level of each of these small molecule compounds. If we take an example, this is a small, tiny chunk of the chemical checker database, because here we can see each row is a compound. So here we see a few hundred compounds, and remember that we have 800,000 compounds. So this is the chemical checker vectors. And as you can see in the classical solution, we would be seeing only this information, which is related to the chemical information. But with the chemical checker vectors, we are actually able to have all this information about each of the molecules. If we can, then what we can do is we can plot all these molecules. So here we are plotting 10,000 compounds into what we call these maps. Each dot is a molecule, and we can see that these molecules are clustered. Molecules that are in the same cluster means that they are similar according to chemical checker signatures. So if they are similar, we expect them to have the same effect. So indeed, if we look here, for example, these molecules target the estrogen receptor 1, or these molecules target the MAP kinases 8 and 9. What is interesting to see here is that some of these clusters are indeed chemically similar, but some of these clusters are not chemically similar. So they are similar, and we can identify that thanks to all these other levels of information that are added into the chemical checker signatures. To put some more examples of why is this important, here we have DNA damaging drugs that are used in cancer. So if we plot these drugs into these maps using the chemical checker signatures, we see that they elicit the same transcriptional responses in human cells. They have the same sensitivity profiles, but their structure is quite different. And having this path allowed the group of Patrick to identify a new molecule with potential activity as an anti-cancer drug. The other way around is also true. So if we look at two groups of different types of antibiotics, we see they are chemically different. They have similar mechanisms of action in the bacteria, but the transcriptional profile that they elicit in human cells is quite different, which is very important when understanding the side effects of each of the antibiotics. So why is all this so important? Because why we need such detailed information? Well, because the chemical space is huge. There are more than 2 million molecules that are available, and we only have information actually for 1% of them, and only 5,000 of these compounds are actually approved or drugs in development. So we need ways to fill in all this information we don't know. So if we take a look at the chemical checker, the reality is that we have chemical information for most of the drugs that are included in the chemical checker, but we don't have so many information at the other levels, especially in the clinical level. So what they did, what Mikel and his coworkers did at IRB Barcelona, was to develop Siamese neural networks. I'm not going to go into detail on how these deep learning algorithms work, but basically they try to impute these gray spaces here. So by using information from targets and from networks, they try to impute for example, what would be the effect on cell assays so that they can fill in all these spaces. This was trained for over a week in a supercomputer center that is actually at Barcelona. It's very nice. It's inside the church because churches are very cold. So that's where they keep the computers to maintain their refrigeration systems easily. So after training this, they were able to fill all these spaces. So now with predictions and therefore allow for much better information about the chemical space. So after this, if we come, we can have some examples here. So if we plot the drugs, the known drugs, we can see that drugs for infectious diseases cluster together, drugs for oncology cluster together, and so on and so forth as expected. But for fun, we can also use other molecules, which is interesting when we want to do assays on, for example, natural plant products. So if we, for example, plot the metabolites instead of the drugs, we see that these are metabolites that correspond to humans, but these are metabolites that correspond to yeast, so they cluster very nicely together. We can even do that for food ingredients. So these are food ingredients that are found in the meat. Instead, these are food ingredients that are found in the coffee. So this is all great, but where does it fit into the Ersilia Model Hub? If we come back to our original task, the chemical checker allows us to have much better and much faster information about the raw data and process data that we feed into our models. So if I can give a brief example about artificial intelligence and a simple artificial intelligence task, this is a growth inhibition assay against E. coli. So we have done the experiments. We have some molecules. We know that this one is active. This one is also active, but these two are actually inactive. So what we do is we encode these molecules into signatures to feed them into the computer. Then we train this model, no? So this model knows which molecule is active and inactive. And when we present it with a new molecule, it will be able to predict whether it is an active or an inactive. As you can see, this molecule is predicted active. It has certain features that are similar to the active ones. So the chemical checker is helping us in this task, basically at these steps. So we are able to use chemical checker signatures instead of traditional fingerprints, therefore doing this process much faster and much easier than with traditional solutions and providing much more reliable predictions. So as I was mentioning, we have already some success stories. The chemical checker was a winning technology in the Dream Challenge last year. We have also done some work in malaria immunology and also in analyzing medical records in Zambia and in Mozambique. And yeah, this is our roadmap. So Ersilia was born in February last year, where we had this initial idea, and then we started working full time on it in September 2020. We were recognized by the Charity Commission in the UK in November last year, and we have already started some partnerships, mostly in Africa. By January, we got accepted this Lancet Global Health paper, and we are applying to grants together with our partners. And we hope that we will be able to launch a prototype of our model hub by next month. We are building our board of trustees to help us guide in our mission. And we expect by this year, we can already submit a paper and include up to 100 models in our platform. And ideally, we would like to have a partnership where we can already validate some of our models. So I think, yeah, the take-home messages are very easy. We are building a repository of artificial intelligence models that is, for the moment, focused in drug discovery. We use the chemical checker as a background technology, which allows us to train high-quality models. And we are a not-for-profit initiative, and we really are very interested into partnering with researchers to help them solve the problems they might have and to help them implement sustainable artificial intelligence tools. So that's us. Yeah, thank you very much. And I'm gonna stop sharing now.
Thank you, Gemma, for the nice talk and introduction. Quite interesting. And first of all, I want to say good luck, since you're about to make public your model hub. So I hope everything goes well.
Thank you.
And I think Miguel had a question.
Yeah.
I think.
No, actually, it was just an applause.
Okay, thank you.
Great. Anyone else that has a question? Okay, we have more people. Yeah, nice. So, I mean, I'm also not in the field of computational and modeling and that stuff. So my question would be of a kind of general, like, if one wants to contribute to Ersilia, is it possible? So
Yes.
kind of like the backward direction.
Yes, that's actually one of our goals. So maybe I didn't mention it clear enough. So the Ersilia platform is fully open source. All our code is available on GitHub. So we welcome developers that want to contribute on one hand, and then on the other hand, we also expect that scientists that have relevant models or even already published that they want to contribute them to the Ersilia Model Hub, they should just contact us, and we will be more than happy to include them in our hub. We are actually preparing some documents to make this easy process. So yeah, nice. That's one of the goals, of course, to just foster collaboration.
And maybe
if I may, I have a question for Gemma also. Yeah, so the old-fashioned approach to this is the combinatorial chemistry, right? So you have a natural molecule and you try different changes in that molecule and see what happens. So your program allows for that combinatorial chemistry in silico, say? So
yes,
you can modify things and predict whether this is going to be better or worse than the original.
Yes. So maybe, so we can develop models to do that. It depends on the type of data that we have, the model that we can train or not. Maybe, I don't know, Mika, you want to add something? If you are
here. So the combinatorial chemistry would be basically the primitive version of what today we call generative models. And generative models, basically, given a seed compound, are able to generate chemical diversity around this one, right? Which is a little bit the idea of the old idea of combinatorial chemistry. So yes, we do have generative models in our hub, and we are planning and we are fine-tuning them towards different activities. So definitely, yes, we do encompass combinatorial chemistry and generative models in our hub.
Okay, thank you.
So I think your question is very related to a question in the chat, which is, in the entry of a compound for an artificial intelligence check on its activity, what if my molecule to be a new entity is designed with, it's a derivative, I understand, using quantitative structural relations, whose chemical information is not available. So it's sorry, I don't understand. So you mean that your new molecule would not have the chemical information yet available?
I think that's what Abdul Basit means. So that he has a new molecule that it's not known anything. I guess some information.
Some information about the
must be
chemical
known. Exactly, but
yeah. So as I was explaining, with the chemical checker, we can try to find similarities at different levels with other molecules. So try to input what you don't know about this molecule, and then make the predictions. Of course, it depends on the data set. So if your molecule is close to the data set that's been used to train the model, the prediction will be highly reliable. If it's not, it will be less reliable, but all the models have confidence score, so you will know whether the predictions we are making are really accurate, or because the molecule you are presenting is very different to everything that is known, is actually not so reliable.
I have one more question. I mean, it's not completely related to drug discovery, but would it be possible, or kind of like an extension of something like this, just for normal like predictions on protein-protein interaction? So not directly on the global health point, but mostly also for, I think many scientists, it's interesting. At some point we face this problem of, is my protein interacting with this protein? Something like this, would this be something you considering to broaden it? I mean, of course, you're quite new, so.
Yes, so not for the moment, we are not so much into the realm of protein-protein, because that requires crystallography in many occasions and many times. But yeah, that's something that in the future we may be including in our database. We just start with the compound, with the drug discovery, because we have the chemical checker resource, and yeah. But yeah, this is something we will tackle in the future, for sure. Okay, I think.
Yeah, I think Abdul Basit.
Yeah, I can. I'm just writing my email. So if you or anyone else is interested, you can just contact us in this email, or you can also, you have our Twitter is actually linked on the Trend in Africa Twitter.
Yeah, and of course, if anyone needs any more information, you can always email us in Trend, and we will bring you in contact with Gemma and the people in Ersilia. Any more questions from someone?
So I was thinking. Hello once again. Yeah, Gemma. So thank you for the presentation. It was really wonderful, and I see, like, it is kind of a very broad kind of spectrum of things that can be done using this AI models. Looking at in like, in the chemical aspect, like using actually this chemical checker. So my mind went to chemical ecology of insects, the vectors of diseases, actually. So there are a lot of different types of chemicals that usually they put for, like, to target some particular insect. So if the information of the receptors of those insects are available, and then maybe the chemical checker can be used, like, to do, like, a test on this spectrum of how this the interaction between the two, if it is possible. And then from there you can be able to design very specific insecticides for, or chemical repellents or something.
Yeah, actually, that's a very good question. Thanks. So the chemical checker was originally developed in the at IRB Barcelona in the field of cancer and Alzheimer's, and we are repurposing it now to the infectious diseases. So this is something, for example, that we are tackling actually in a project that we are probably starting about insecticides. So yes, definitely not right now straightforward. We are just actually working on this to repurpose the whole chemical checker signatures for infectious and vector control and all these things. So yes,
that'll be fantastic. Yeah,
that's something we will tackle indeed. Yes.
Oh,
thanks
that's really fantastic. All right then.
So,
are there any questions? So I think there aren't any more questions. So I would like to thank you guys for being here with us, and you, Gemma, for the nice presentation. And
thanks
well,
to you.
Okay, yeah, and I hope for many collaborations in the future, and we will be in touch, I guess.
Yes, thank you so much. Thanks everyone for coming, and yeah, don't hesitate. So I think this is being recorded, so we'll have it in the Trend in Africa and also in our YouTube channels. So if you want to forward this to other people in your organizations that may think it's useful, please do so, and yeah, contact us with any more questions or proposals that you have. Thank you so much for your time.
Thanks to you, and bye.
Thank you everyone for following up. Have a nice evening or whatever time zone everyone is at. Bye.
Bye.
Bye.